The next talk is by Akihiko Yamamoto, entitled A Delicate Balance of Mating Preference in Drosophila melanogaster. Thank you.
I thank organizers who gave me a chance to present my research. I'm interested in the genetic basis of speciation. I talk today about my finding on genetic background and the involved biological functions of premating isolation of two races of Drosophila melanogaster. Our favorite material, melanogaster, has two races. One is cosmopolitan, to which most laboratory stocks belong, and the other is African, which was first reported 2 decades ago from Zimbabwe. The key character is African females avoid mating with cosmopolitan males. After 2 decades of discovery, we still don't know detailed genetic background of this premating isolation, even though it is happening in Drosophila melanogaster, one of the best material for genetic analysis because of prevalent inversions in the African population. While most of the efforts focused on African females, I took a new strategy to analyze cosmopolitan males. I analyzed genetic basis of variation of interracial mating in cosmopolitan males, then used the information to investigate the biological function involved in the premating isolation. First question is, what is the genetic basis for variation of interracial mating in cosmopolitan males? Our laboratory, as a creator of DGRP, it is natural to me to utilize this fantastic tool. DGRP comprises of 205 isofemale lines from Raleigh, North Carolina. They are highly inbred and deeply sequenced. They belong to cosmopolitan race. DGRP is essentially a converter of phenotype to candidate genes. Start of my work was to assess phenotype of DGRP. This is a histogram of 205 DGRP lines for their copulation ratios with G30 females. I use G30 as a representative of African race and copulation ratio for mating success through this presentation. DGRP as cosmopolitan, majority of the male has difficult experience with G30. A half of DGRP lines never reached copulation. On average, 4% copulated in 2 hours, but some lines did up to 30 to 40%. So quite a variation there. As a reference, G30 males, only males of the African stock copulated more than 80%. Publicly available DGRP pipelines converted these phenotypes to candidate genes at the brink. We identified more than 500 SNPs significantly associated this phenotype, and it's corresponding to more than 300 genes. I selected 53 promising genes with various reasons for further analysis. And 42 genes, about 80%, were confirmed by RNAi interference mediated gene knockdown test. I explain detail of this test soon. As a control, I tested random 9 RNAi, and only one was weakly significant. I used 3 drivers for the confirmation test. all of which drives ubiquitous expression of transgenes under US sequence, but in various degree. I present here the result of 27 genes, which I tested with all 3 drivers. Top one, ubiquitin 156, is a weak driver, which produced viable offspring with all RNAi. Middle and bottom, ubiquitin and actin, are stronger and kill some combinations, presented here as a blank. First, pay attention please that gene knockdown males did mate better. Their copulation ratios were higher than the controls and often very significant. Only few cases marked by red are the suppression. This means that the normal function of these genes is to make males ugly. Second point I want to emphasize is the weak driver manipulated the phenotype most. I explain of this driver strength effect further using the 12 genes which gave viable offspring with 3 drivers. Left panel shows driver strength assayed by luciferase activity. Right panel shows copulation ratio of 12 genes with 3 drivers. When drivers went stronger, the copulation ratio depressed. So gene knockdown effects are on delicate balance. Then I wondered that those gene knockdown effects were specific to African females. For further analysis, I focused on the 4 genes of the largest effects in the confirmation test. Black bar is a control, and 4 knockdown males were in gray. All of them are highly significant with G30 females, as I already told. How about cosmopolitan females? I used Oregon Samarkand hybrid as cosmopolitan female, and they mated with gene knockdown males more, though not as striking as G30 female did. So the gene knockdown effect on males are not specific to African females. It is a little disappointing, but remember that these 2 races belong to the same species, so we should not be surprised. Soon, I recognized this as a new opportunity. When cosmopolitan females detect the gene knockdown effects, laboratory stocks with various sensory mutations, they are cosmopolitan, can be used to ask how females sense the knockdown males. First, I tested white-eye mutant Canton SB. They are partially blind, but they did a good job to detect the gene knockdown males. When their antennae were surgically removed, advantage of gene knockdown males were gone. And triple mutant of olfactory receptors, IR8A, IR25A, and ORCO, they are completely smell blind, but they even unveiled the inferiority of the knockdown males. So I concluded that female used olfaction to detect the gene knockdown males. How do these genes control male smell? After dozens of tissue-specific drivers, and Oenocyte-specific drivers gave the answer. Prom-E 800, reported by Britta and others, has very specific expression in Oenocytes and little elsewhere. when the 4 genes I've been using were down-regulated in oenocytes, male copulated much better than the control. So, oenocyte is the tissue responsible for the gene knockdown effect. Oenocytes synthesize cuticular hydrocarbons, and they are exported to the outer layer cuticles as chemical signals, smell and taste of fly. So, cuticular hydrocarbon profiles of the gene knockdown males are altered. That's the next question. We examined our gene knockdown males. I present CHC profiles of the gene knockdown males with Ubiquitin 156 as driver. Average of the 4 genes was shown against the control. Because these 4 genes reacted similarly through my various analysis. Cuticular hydrocarbons are in order of abundance. From this picture, I noticed that many of the depressed CHCs in gene knockdown males are methyl-branched. I highlighted all methyl-branched CHCs, and majority of them are decreased in the gene knockdown males. These observations don't prove that these are fat female sensed, but it fits nicely to the other findings, such as delicate balance of gene knockdown effects or their expression in oenocytes. Now I summarize my talk. The cosmopolitan males' characters of interracial mating are regulated by multiple genes, which controls their olfactory signals, possibly through cuticular hydrocarbon composition. Now we have better understanding of genetic control and biological functions involved in the pre-mating isolation of the 2 races, at least as the cosmopolitan side view. As I finish my talk, I present scenario of the 2 races evolution according to my finding. Africa is the homeland of melanogaster. They invaded to other continents, maybe following human migration. In order to adapt to new primates, cosmopolitan population adjusted their CHC profiles and accumulating methyl-branched cuticular hydrocarbons. Now, cosmopolitan males smell bad, and African females treat them discriminatorily. So the pre-mating varies. I acknowledge all the help of colleagues in Mackay lab and Anholt lab of North Carolina State University, especially Richard Lyman, who is a key person of our DGRP project. Without him, we don't have DGRP as it is, and without DGRP, my work wouldn't start it. Thank you for your attention.
These are the same flies that there's a story of Jerry Coyne's where you can put the African flies in with the cosmopolitan ones, and it makes the effect go away of the mating isolation. You can change the hydrocarbons on the outside of the fly and make them attractive.
No, I didn't do anything. I just assessed the profile.
The microphone.
I didn't do anything. I just we assayed the cuticular hydrocarbon profiles as a total. We didn't change. And actually, that's a very subtle change, maybe, because weak drivers affecting most. So such manipulation may be too much effect. That's my guess.
Okay, thank you. Thank you very much.